athletes who don't cheat can win once in the world. Boy, we see astounding performance yesterday at Marathon in Austin, Texas. Here's some overheads.
Kenyan runner Glycogen had been in the lead for nearly 26 miles. The finish line was within sight when this happened. Overcome by exhaustion, Glycogen fell onto her hands and knees, but kept going.
What is happening is that suddenly not more. Suddenly there was a deprivation of energy, and she had to walk.
When the medical team rushed to help and offered her a wheelchair, she refused. Glycogen had still managed to come in third. Conley rated her after the race.
You ran the bravest race and crawled the bravest fall I've ever seen in my life.
Glycogen couldn't recall those final moments.
But the last 2 kilometers, running always, you have to keep going.
Conley bumped up her cash prize. He says it was the least he could do.
I've never heard of her crowds that high. Should have been went back. It's like she won the race. She is the defining moment of that weekend for us.
An image of defeat turned into triumph. And
So actually what happened here is what is called in sports medicine language is hitting the glycogen wall. So those who are into intense sports, if they are not taking a proper carbohydrate meal before their big race, they may have this sudden deprivation of glycogen and they may lose their abilities to even move their limbs. That is what happened with her. So we require, for maintaining of our blood sugar levels and for punching of our muscles, we require glycogen. So what is glycogen? All of you know. Can anyone tell me? It is a homopolymer of glucose. It is made up of amylopectin, amylose, manpolo, that are components of starch. You remember? What is amylose? What is amylopectin? What is amylopectin? Which one of them is branched? Amylopectin is branched and amylose is not branched, it is unbranched. So there are two components or two forms of starch. And what is the difference between amylopectin and glycogen? So glycogen is more frequently branched. 10 to 12 units ke baad there is a branch, while in that it is 12 to 20. So it is less branching. Okay? So animal starch is glycogen. And why do we need to store glycogen in form of glycogen? Why do we need to store energy? Why not simply glucose? Very good. One is, both of you are correct. One is that the osmotic pressure of glucose is very high. You cannot store it like that, the cell will burst. The second is, glycogen is very compact. Okay, so glycogen is stored at two places mainly in our body, and these two places are liver and muscle. One more thing here which liver and muscle are right. So the percentage of glycogen which is there in the muscle is a little bit higher. I'm telling you just the opposite. So the percentage is higher in liver, but overall glycogen is higher in the muscle. It is because we have more muscle mass. So problem with glycogen muscle, or the characteristic of glycogen muscle, is that it is available only for muscles. it will not contribute to the blood sugar levels. Why? Because the enzyme glucose 6-phosphatase is absent in the muscles. So the contribution of glycogen to the blood sugar levels, it will be possible only with the liver glycogen, though it is less in quantity. You have more quantity of muscle glycogen. However, percentage wise, liver has more glycogen. Okay, so two places in which it is being stored. So if there is any disorder which is related to glycogen metabolism, these two places will have the main manifestations. Okay. So this is what I was trying to explain here, that liver glycogen can contribute to blood glucose levels in the serum. However, in muscle it will be utilized only for the muscle or muscular activity. There are reducing and non-reducing ends of glycogen. So the non-reducing end, which one will be the non-reducing end and which one will be the reducing end? What is the bond which is there in glycogen? Alpha 1, 4. So where we call it is a reducing sugar. How a sugar is a reducing sugar? With the help of which group? The functional group. So here the functional group is at what position? At the first position. So wherever it will be the fourth one, which is in the end, which is a non-reducing end. So non-reducing end will be more frequent in glycogen and reducing end will be less. They are infrequent. So coming to synthesis or formation of glycogen. Now glycogen is formed by the glycogen formation requires activation of glucose to UDP glucose. UDP glucose formation is required. Now how it is formed? Initially you will start with phosphorylation of glucose. So glucose can be phosphorylated by hexokinase or glucokinase. The difference I will tell you when I am telling you about glycolysis. There are certain people who are sleeping in the class. They are coming because of the low attendance. रात को पढ़ते हैं, I hope all of them. पढ़ते हो रात को? कितनी देर तक पढ़ते हो? बताओ. रात को कितने बजे तक पढ़ते हो? Whole night? नहीं? तुम बताओ प्रज्ञा. कितनी देर तक पढ़ते हो? तो जब कब तक तुम्हारा मन करता है? In a routine night. Not up to 12:30. Okay. And how much of sleep is required for a healthy, active adult? It is actually very individualistic. Please sit down. So 6 hours on average. Now this glucose 6-phosphate is converted into glucose 1-phosphate with the help of phosphoglucomutase. So what raw material you require is glucose converted to glucose 1-phosphate. And you ultimately need to form the activated form. So this is done with the help of this reaction, which is the enzyme is UDP glucose pyrophosphorylase enzyme, in which glucose 1-phosphate and UTP they react and form UDP glucose and pyrophosphate is released. Now this will move in the forward direction. Why? Because pyrophosphate on converting into inorganic phosphate will release a lot of energy. So it will move in this forward direction. Okay? So UDP glucose आपका from UTP and glucose 1-phosphate it will be formed. Now we also require a primer in this. Like for DNA synthesis you require a primer. Here also a primer is required. Why a primer should be required? This primer is called as glycogen and it has certain tyrosine residues which can actually add glucose 1-phosphate units together and form at least a 7-member chain. So the initial formation of glycogen will be with the help of not glycogen synthase enzyme but glycogen itself. So glycogen will form a short chain. And why this short chain is formed? Why not glycogen synthase can form from the zero? Why not it can take two glucose molecules and attach them and then start making a chain? It is because glycogen synthase affinity is very good. Affinity for the substrate is low. That is why you need at least some amount of chain for the activity to start. Okay, so glycogen is required as a primer for glycogen synthesis. Please don't talk like that. खड़े हो जाओ बच्चा. खड़े हो जाओ. नहीं तुम नहीं. साइड हो. ये लड़. हाँ. खड़े हो जाओ. आ जाओ. यहाँ आ गया. यहाँ से नहीं प्रखर को डिस्टर्ब करते हुए आओ उधर से. Not the whole row. And all of you who are trying to sleep here, I have no problems with your sleeping, but we will not give you attendance like this. Making cat calls in the class. Stupid. Come, sit in the front. Respect नहीं है, मत हो कोई बात नहीं. I don't care for your respect, but you have to follow the decorum. Very bad. So glycogen synthase, the enzyme, remember it will act on the primer at the non-reducing end. So it will be adding to the non-reducing end. So at the fourth position, it will keep on adding more and more chain. Okay. So this is how glycogen synthesis occurs. Here activation occurs, there is a primer which is involved and a chain is formed. Now this chain, when it becomes up to 11 residues long, then a branch point is introduced. So for introducing of the branch point, there is another enzyme which is a branching enzyme, that is amylo 1416 transglucosidase enzyme. And what it does is, from a 11 polymer long chain, it will take 6 residues and attach them at some other chain point and introduce a 1,6 bond. So it is removing 1,4 bond, making that chain go to some other place and get it attached in a 1,6 point. So that will be the branch point. Okay. Now, regulation of glycogen synthase. So this is how the whole glycogen will be formed. They will be adding with the help of glycogen synthase, and branching enzyme will introduce more branch points. Okay, so you have now a fully formed glycogen. But how will it be regulated? How the synthesis will be regulated? The synthesis of glycogen is regulated with the help of enzyme glycogen synthase. So glycogen synthase enzyme, remember, is active in the dephosphorylated form. Now why it is so? That one enzyme will be active in the dephosphorylated and the other in the phosphorylated form. Actually, most of the metabolic activity, if you look at glucose metabolism, insulin and glucagon. So well-fed state mein you have insulin, and in the starving state when you require glucose, do you require to synthesize glycogen then? In a well-fed state only glycogen will be synthesized. When the body is starving, then glycogen synthesis is not required because it is using up all that glucose which could have been utilized by the brain cell, by the WBCs, RBCs, etc. So for that it has to be inhibited. So whenever there is a decrease of glucose, there will be release of glucagon or counter-regulatory hormones to insulin. So there are many of them, like growth hormone, catecholamines. So one of them is glucagon, which is the main one. So it acts through a second messenger. This is the second messenger for glucagon, cyclic AMP. Okay. Have you been taught second messenger? That has already finished. So you know what is cyclic AMP? What is cyclic AMP? It is the bond is formed between, the cyclic bond is between jacket pattern, because this is already covered. Okay, between the third and the fifth in the ribose molecules. Read that. It is formed from ATP. So from ATP, with the help of adenyl cyclase, you will get AMP, and this AMP will be further converted to 5' AMP with the help of phosphodiesterase. Okay. So this will be inactive. So when we have increase in the cyclic AMP levels, it may be because of various counter-regulatory hormones. There are many counter-regulatory hormones to insulin. What is the function of insulin? To decrease the blood glucose levels. And the counter-regulatory hormones, there are many, because if one mechanism fails, the other will function and the body will still survive. So we have many of them, and they will function through cyclic AMP. Mainly we are talking about glucagon here, and it will act on a protein kinase. So this protein kinase is made up of two units, catalytic and a regulatory subunit. So two catalytic and two regulatory subunit, C2R2. This will bind with cyclic AMP and the regulatory subunit will be removed. So now you have catalytic subunit, that is activated protein kinase. Okay. This activated protein kinase will now, what is the function of kinase? To add phosphate. So it will add a phosphate and make it inactive. So mission accomplished for the counter-regulatory hormones, because they want to stop glycogen synthesis. They want to increase the glycogen breakdown. And which enzyme will be for that? Glycogen phosphorylase enzyme is there. So what do you think, glycogen phosphorylase will be active in the dephosphorylated form or the phosphorylated form? इसको देख के बताओ. This is inactive in the phosphorylated form. What about glycogen phosphorylase? What it should be? Very good. It will be active in the phosphorylated form. Okay. So this inactivation will occur. Second way in which it is doing is, if you see, slightly in a different way, which is a longer way, is that it will act, the inhibitor will be inactive and it will be phosphorylated. It will inhibit the phosphatase enzyme also. So two ways by which it is doing. One is directly acting on the enzyme glycogen synthase by phosphorylating it, and by inhibiting the phosphatase, which is acting and converting it into active form. So in two ways it is trying to regulate glycogen synthase activity. Have you understood? Have you understood this protein kinase? Here it will activate the inhibitor, which is inhibiting the activity of phosphatase. ठीक है? Glycogenolysis. Now glycogenolysis, it is present in various places with slight differences. There are slight differences, but the main part is that the characteristic of muscle phosphorylase, you remember, it is in the active form, it is a tetramer and has 4 molecules of pyridoxal phosphate. In the inactive form, it will simply be a dimer, it will be broken down, and the molecules of pyridoxal phosphate will also be less. What it does, it will break down glycogen with the help of phosphate. So to each glucose which is coming out, a phosphate is added, and the ultimate product is glucose 1-phosphate. What was added? Glucose 1-phosphate was activated. Here also you will get in the end glucose 1-phosphate. This glucose 1-phosphate can then further be broken down. It will be converted into glucose 6-phosphate and then that will be broken down with the help of glucose 6-phosphatase enzyme, which is absent in the case of glucose 6-phosphate is absent in case of muscle. So it will not function. Now, at the branch point, this phosphorylase will not function. So at the branch point, you require something else. When 4 molecules of sugar are left at the branch point, this is a whole chain in which many residues are there, and here you have a branch point in which 4 residues are there. So what it does is this enzyme, this is a debranching enzyme, it will break here. So a tripeptide is removed. Now you have a single peptide. This single peptide is broken down and you will get glucose. So here you will get a single glucose residue. So in 2 parts it will be doing this. The step 1 is that it will be taking the outer 3 glucosal residues attached at the branch point, that will be removed, that is the first step. And then the same polypeptide will have 1,6-glucosidase activity. So at the branch point it will break without introduction of a phosphate group. So whenever a branch point will come, you will get free glucose. So end products for glycogen metabolism will be actually 2. Glucose 1-phosphate and free glucose at the branch point. So both of them together are called as debranching enzyme. Now again the same thing here, that when do you require to break down? In the starvation or when the state is not a well-fed state. More cyclic AMP is there in the cell because of more glucagon. So more cyclic AMP means the protein kinase will be activated. The same protein kinase. Here this protein kinase will activate phosphorylase kinase. So not directly acting on phosphorylase but on phosphorylase kinase. And this phosphorylase kinase will be activated. It will phosphorylate the phosphorylase enzyme and make it active. So not directly. And why most of the reactions will have this indirect kind of control, it is to fine-tune the control. If there is only one protein involved, then something may go wrong. So there are 2-3 proteins involved. There may be other mechanisms also of which we are unaware. One of them I will tell you. So there are many mechanisms by which the body is actually dealing with glycogen, and few are known to us. The other mechanism or the second pathway of glycogen degradation is with the help of enzyme alpha-glucosidase. This enzyme, acid alpha-glucosidase enzyme, is present in the lysosome. So there is a separate pathway in the lysosome. Okay? The third point, which is very interesting and actually it is. So I have told you the ultimate product we require from glycogen breakdown was glucose. But this glucose has to be converted with the help of, first product was glucose 1-phosphate, it was converted to glucose 6-phosphate with the help of enzyme phosphoglucomutase. Then it required glucose 6-phosphatase enzyme. So this enzyme, glucose 6-phosphatase enzyme, the active component or the enzymatic component is embedded in the endoplasmic reticulum wall. So it is inside the ER that you have the active component. That means glucose 6, which is the glycogen is outside the endoplasmic reticulum, it has to be taken inside. So what is required? You require a translocase. So a translocase protein will bring the glucose 6-phosphate from the cytosol to the endoplasmic reticulum, and inside the endoplasmic reticulum it will be broken down into glucose. So कितने steps हो गए इसमें? Phosphorylase enzyme में क्या किया? Breakdown of glycogen phosphorylase enzyme will break it down into and at the branch point into free glucose. Then if this glucose 1 phosphate will be converted into glucose 6 phosphate, and then 6 phosphate will be taken inside the endoplasmic reticulum, where it will be broken down by glucose 6 phosphatase enzyme, which is having the enzymatic component inside the endoplasmic reticulum. The other way, which is also called as glucophage, is that this will be broken down at the lysosomal level. This is related to a disease, so it is important to know. Now, there are certain disorders which are related to glycogen storage, and these disorders, this is a mnemonic for them. There are many others. So there are many more in the book, so you will see many more are mentioned. But if you remember first few of them, that is fine by me. So Von-Geiger's disease is type 1. It has 2 components, I will tell you. Then comprase is the lysosomal degradation. That enzyme is deficient. Cory's, Anderson, structure defects are there. McArdle's, M, remember M for muscles, so muscles are involved. Hers is very similar to Von Geiger's disease, low intensity though, and Tory's. So each of them you will have to remember for your short note probably. So the 0-1 glycogen synthase deficiency, in this hypoglycemia is there. This I will not be covering here, you can read. Von Geiger's disease is there, in which glucose 6 phosphatase deficiency is there, or the transporter is absent. So there are 2 types of Von Geiger's disease, 1A and 1B. Okay. And comprase, lysosomal acid maltase or alpha acid glucosidase is absent, which is also called as acid maltase. Limit restenosis. What is dextrin? What is dextrin? Breakdown product of, so breakdown product of starch is dextrin. Limit restenosis means that the muscle debranching enzyme is, or the liver debranching enzyme is not proper, and when you see under a microscope it will give that kind of structure. So limit restenosis can again be various subtypes, you need not remember that. Then Anderson's disease or amylopectinosis. Amylopectin क्या वो? Starch, which was having less starches. So here the glycogen which is abnormal is actually resembling amylopectin. It resembles amylopectin. So that is why the name is amylopectinosis. Then muscle phosphorylase, or what is McArdle's disease? MM, remember that. Muscle phosphorylase means the muscles will have low energy because there is no glycogen. I told you, hitting the glycogen bond. So that will happen every time there is some intense activity. And whenever such a thing happens that you are actually pushing your muscles to work but you don't have energy, muscle degradation can happen. So what is called as myoglobinuria can happen. Myoglobinuria में कैसा यूरिन आता है? Brownish, reddish, or what is called as a Coca-Cola colored urine will come. There are many more, so you decide how much you can read. I will be telling you only about the main ones. So coming to the Von Geiger's disease. So the first one, Von Geiger's disease, in which there is a deficiency of glucose 6 phosphatase. It can have various features which are coming to the case report. It will present as abdominal distension, vomiting, lethargy, and there may be death also. You can have a family history of similar episodes in the family, that someone dying because of some liver in small childhood only. And consanguineous marriages are also very common as a history. So what is the biochemical basis for that? I will try and explain. So whenever there is glucose 6-phosphate which is there, you are taking it out inside the cell and it has to be broken down. Now various ways of doing it. If there is excess amount of glucose 6-phosphate in the body, how it will be utilized? कैसे वो utilize कर सकता है? One way is that it will enter glycolysis and will enter, so it will form pyruvate. This pyruvate which is formed, which will now be in excess. So this excess pyruvate, some part will go to TCA cycle by forming acetyl-CoA and some part will be converted into lactic acid. So for the first feature which is there is presence of lactic acid in excess amount. So what is called as lactemia will be there, or lactic acidosis will be there because it is an acid. So the first feature here is because of the increase in the pyruvate, it is lactic acidosis. Now the second feature which is related to this is increase in the uric acid concentration. Now lactic acid competes with uric acid for excretion. Uric acid is a waste product. The body is making it and is throwing it away in urine. But if lactic acid is also present, which is not normally present, there is a competition between uric acid and lactic acid for excretion, and body will excrete less of uric acid. So there will be hyperuricemia. Okay? So दो features हो गए biochemical. There will be hypoglycemia. It is because whatever glycogen is there is not being available to add to blood sugar levels. So hypoglycemia is obvious. Okay? Now this glucose 6-phosphate is producing a lot of acetyl-CoA also, because whatever pyruvate was being formed, it is going to TCA cycle. So a lot of acetyl-CoA is there. This acetyl-CoA can form various things. So one of them is formation of de novo fatty acid synthesis. So more fatty acids will form, more triglycerides will be formed, there will be increase in the triglyceride levels in the blood. Okay. So the features, biochemical features now are, the first and the foremost which the patient will present with is hypoglycemia. The second one, there will be lactic acidosis. Then there will be uric acid increase. And lastly, there will be increase in the blood levels. So these are the biochemical features which will be there in von Gilkes disease. And they are asked in the Viva. In the Viva, they will ask you what all happens in von Gilkes disease. Von Gilkes disease has a characteristic moon-like face or doll-like face, and they have a protuberant abdomen. The protuberant abdomen is because of hepatomegaly. So massive hepatomegaly is part of this, and if you see the picture, I think I do have protuberant abdomen picture. So this picture will be like this. The child will have a doll-like face and protuberant abdomen. If these two words are written, then you can think of von Gilkes disease. So what all biochemical features will be there? Can anyone enumerate? Hypoglycemia will be there. Lactic acidosis will be there. Hyperuricemia will be there, and hypertriglyceridemia will be there. So initially they used to find out or know the cause with the help of liver biopsies. Nowadays they are not doing that. They can get glucose gene sequencing done for the genetic profile. So in a normal person, they will not be stained so that there are large vacuoles of glycogen there. The hepatomegaly is evident here, and you can stain it and see whether it is glycogen or not. Now it is not done. Then because of the disturbed lipid metabolism, there is lot of cholesterol, lot of triglyceride there roaming in the blood vessels. So some of it will be deposited. So deposition of cholesterol, it gives a yellowish color and this kind of marks in the skin. You might have seen in the eyebrows, a lot of people, elderly people, they have. So this is deposition of fat, and it is because of excess of cholesterol in the blood circulation. Protuberant abdomen because of hepatomegaly. This I think we'll skip. This you already know. What is the treatment? The treatment is to avoid hypoglycemia. So how can you avoid hypoglycemia? You have to give something which will give a constant flow of glucose. So the constant flow of glucose is possible if you give cornstarch. Cornstarch is digested slowly and it will slowly release glucose. So every 3 to 4 hours that patient is given cornstarch depending upon the weight of the body. They are given cornstarch meals. So main meal or main source of energy here will be carbohydrates, and they have to be given cornstarch even during the middle of the night, because sleeping during the whole of the night is not possible. They will undergo hypoglycemia and may die, may not wake up only. So they have to be given cornstarch in between night also. So it disrupts their normal living pattern, So here if you see this kind of MCQ will come. Fasting blood sugar levels are low, hypertriglyceridemia is there, lactic acidosis is there, hepatomegaly is there, venomegaly may be there, and there is increase in the glycogen content. So what is the diagnosis? It is von Wieck's disease. So if you see lactic acidosis, hyperuricemia, hypertriglyceridemia together with some hypoglycemia also mentioned and glycogen disorder, you should think of von Wieck's disease. A 2-year-old boy presenting with fasting hyperglycemia, hypoglycemia गलत है, hypo होना चाहिए, doll-like faces and thin extremities. This is a protuberant abdomen. So this will be type 1 glycogen storage disease. Coming to the second one. The second one was because of lysosomal disorder. It was a lysosomal disorder. Okay, so here the features are, there will be cardiomegaly and there will be floppy baby. The baby will not be, the muscles will not be able to maintain the tone, because for that also it requires a constant flow of glucose, which is not possible in Pompeis. So the floppy baby or hypotonia of the baby is the line which you have to look for. Now that is for MCQs. For MCQs, you will get a very characteristic case in which all the features will be there. Otherwise, Pompeis can be of various types. It can present in children and adults, it can present in infants, and depending upon the activity, in infants it will be slightly more severe, in adults and in children it will be less severe. So for this goes for most of the genetic disorders, they have a varied array of manifestations which are not possible otherwise. You cannot say yes, this is there and these kind of features are there. It is not like that. It will be a whole array of manifestations. So if you try and pick up only the characteristic one, remember hepatomegaly, cardiomyopathy, and microglia is also a feature here. So if there is a floppy baby also mentioned, 100% it is Pompe's disease. So you have to remember or look for the keywords, which I think you are using very frequently. क्यों देवांश? You using it ना? तुम नहीं कर पाए, और लोग तो कर रहे. Okay. छोड़ दे, या एक और केस बता दूँ. Rest you read yourself, and maybe in the class for next 5 minutes we will discuss in the first 5 minutes. So I want you to know about just one more case, and then I will not touch this again because that you have to do it yourself. ये myocardial है. अभी तक हमने जितना भी करा उसमें liver was also involved. In myocardials, liver is not So here you will have a very characteristic feature that there is intolerance to exercise, muscular weakness are there, and episodes of rhabdomyolysis are there. Okay, and why is that? Why rhabdomyolysis should be there? In a normal working muscle cell, there will be release of glycogen. This glycogen is only for the muscle, for its own activity, not to contribute anywhere. Why? Why only for the muscles and not contributing to glucose? What is absent? Glucose 6 phosphatase enzyme is absent. Where is glucose 6 phosphatase enzyme located in the cell? In the endoplasmic reticulum. So here there is missing myophosphorylase, so glycogen cannot be broken down. So what happens? In a normal muscle activity, you will break down glycogen, you will receive more ATP, and this ATP can then be utilized. But if glycogen is not being broken down, shortage of ATP will ultimately occur and there is injury to the muscle fiber. So rhabdomyolysis will occur, or what is called as, it will manifest as myoglobinuria. This myoglobin will be excreted in urine, and it will not only create a discoloration of urine which is akin to blood because it can be confused with urea, it will also damage the kidney. So myoglobin will damage the kidney, acute renal failure may occur. So this is what, if there is a muscle injury, there will be, the kidneys will also be damaged and there will be release of myoglobinuria, what is called as a Coca-Cola colored urine. I think I'll stop here. I will share all the slides.